Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive in, don't forget to like, subscribe, and share this podcast with your friends and relatives. It really helps support the Sand Science Experience. Today, we're embarking on a fascinating deep dive into how science has dramatically moved forward in understanding and treating type 2 diabetes mellitus, or T2DM. Our journey begins with a landmark paper from 2007 that first really set the stage for this field. It gave us that foundational understanding of how a specific mitochondrial protein, UCP2, contributed to diabetes.
It really was a crucial first map, you could say, showing us the big picture of how things worked at a pretty fundamental cellular level. I mean, back then, simply identifying UCP2's role, that was the significant, groundbreaking step. It offered a completely new angle on a really complex disease.
Right. So fast forward to today, and we have this remarkable new study that takes those early ideas much, much further. This research, and it builds directly on that 2007 foundation, uses powerful new techniques, things they just didn't have back then, to reveal the intricate details, the dynamic processes that were, well, completely hidden almost 2 decades ago.
Exactly. It doesn't just describe the system like a static snapshot. It shows us how it actually functions, how it adapts over time, and how it connects to broader processes like overall health and even aging.
And it's interesting, the context here.
Yeah, what's truly interesting is how the field has grown. Sometimes the very latest work doesn't always circle back and explicitly reference those early influential papers. And indeed, the most recent paper on a related topic doesn't actually mention the 2007 study, which frankly seems like a bit of an oversight given how important it was.
Yeah, that's a shame.
But the second most recent paper, the one we're focusing on today, from 2025, it truly shines because it explicitly evolves that foundational knowledge. It really demonstrates just how much our understanding has, blossomed since that initial discovery. It connects the dots beautifully.
Okay, so let's unpack this journey then, starting with that pivotal 2007 paper. It explored the role of uncoupling protein 2, UCP2, in diabetes. At that time, UCP2 was already sort of on the radar, wasn't it? For its ability to reduce ATP production and, critically, inhibit insulin secretion by pancreatic beta cells.
That's right. And what's foundational here is that the researchers in 2007 weren't just, observing. They were actively investigating UCP2 as a potential therapeutic target for diabetes, a direct intervention point. This mitochondrial protein, you find it in various tissues, but importantly in pancreatic islets, was known to dissipate the electrochemical gradient. That's the gradient that normally drives ATP synthesis.
ATP being the cell's energy currency.
Precisely. So UCP2 acts like a bit of a leak, letting that potential energy dissipate instead of being used to make ATP efficiently, which of course impacts the cell's function, like insulin secretion. And it was understood to do this differently from its more famous cousin UCP1, which is more about generating heat.
Okay, so if UCP2 was this kind of energy leak, how did they actually test its impact? What did they do experimentally?
They used a pretty clever genetic tool for the time, an antisense oligonucleotide to UCP2 called UCP2-AS. Essentially, think of it as a molecular message telling the cells to make less UCP2 protein. Just dial it down temporarily.
Right, dialing it down.
And they administered this short-term treatment to 2 different animal models of type 2 diabetes. One group was Swiss mice, made obese and diabetic with a high-fat diet, kind of mimicking a common human situation. The other was OBOB mice, which are genetically prone to obesity and diabetes. So 2 different angles on the disease.
And the results. What did dialing down UCP2 actually do? This is where it got interesting, right?
Well, absolutely. To their surprise, actually, this treatment didn't just lead to a significant improvement in hyperglycemia, meaning better blood sugar control.
Which you might expect if it affects insulin secretion.
Exactly.
Right.
But the improvement wasn't just due to better insulin secretion. It also involved an enhanced peripheral insulin action, better insulin sensitivity out in the body's tissues.
Wow. Okay, so 2 major benefits from this one intervention. That's huge. Can you break down the mechanisms they figured out? How did less UCP2 lead to these 2 effects?
Sure. So when they look closely at isolated pancreatic islets, those clusters containing the beta cells partially inhibiting UCP2 led to an increase in the ATP content within the cells, more energy available.
Right, plugging the leak.
Plugging the leak, exactly.
Yeah.
And with more cellular energy, the beta cells showed significantly increased glucose-stimulated insulin secretion, or GSIS, and made it really clear that UCP2 acts like a sort of switch, or maybe ender, controlling how much of that electrochemical energy actually gets channeled towards making ATP for insulin release. Less UCP2, more ATP, more insulin response.
Okay, that explains the insulin secretion part. What about the other benefit, the enhanced peripheral insulin action? Where did they see that happening?
That was another really fascinating piece. The study found that inhibiting UCP2 expression significantly improved insulin signaling, specifically in adipose tissue, fat cells.
Just fat cells, not muscle.
Surprisingly, not significantly in muscle, according to their findings. This suggested a really quite specific localized effect at the molecular level. It showed UCP2's influence wasn't just a blanket effect everywhere, but perhaps more critical in certain insulin-sensitive tissues like fat. Like fixing that leaky tap had a bigger impact in, say, the kitchen than the bathroom.
So the 2007 paper really did lay crucial groundwork, identified UCP2 as a target, sketched out some key mechanisms, but like all great science, it raised more questions than it answered, didn't it? What were the deeper dynamics, the ongoing processes? And critically, how could this be turned into a real therapy?
Exactly. It set the stage beautifully.
And that really leads us perfectly into the 2025 study. As you said, it consciously builds on that foundation, taking those initial ideas and just propelling them so much further. It fills in those dynamic details that we're missing.
Precisely. This is where the story truly comes full circle, moving from that more, let's say, static observation of what UCP2 did to a really dynamic, in-depth view of its function, its adaptation, and these unexpected links to broader things like health and aging. And crucially, this later research pushes us to view type 2 diabetes less as just a metabolic issue, a blood sugar problem, and more as a disease deeply tangled up with chronic inflammation.
Okay, so let's dive into that 2025 paper. It reframes T2DM as an inflammatory disease, focusing on intense autoinflammation, self-inflicted inflammation right inside the pancreatic beta cells. And they found this protein, ostin, is significantly elevated there.
Hugely elevated, yeah. Esting is basically a key sensor for danger signals inside the cell, often related to misplaced DNA, and it triggers immune responses. Finding it cranked up in T2DM beta cells was a major clue.
So what did they figure out was actually driving this autoinflammation? Where does UCP2 fit into this new picture?
This is the core finding. The researchers clarified that UCP2 plays a key role. An inflammatory molecule, interleukin-1 or IL-1, actually pushes beta cells into this autoinflammatory state through a specific pathway, the UCP2 MTD nasting axis.
Okay, UCP2, mitochondrial DNA, and STING. So it's a specific chain reaction.
Exactly. They describe it as an inflammatory response mitochondrial damage cycle. It's a vicious loop. Basically, IL-1 causes damage to the mitochondria. This damage leads to mitochondrial DNA, mtDNA leaking out into the cytoplasm where it shouldn't be.
And that loose mtDNA triggers STING.
Precisely. The mtDNA activates the STING pathway. STING then activates NFP, which is like a master switch for inflammatory genes. This ramps up the production and release of more IL-1, which then causes more mitochondrial damage, releasing more mtDNA, activating STING again, and round it goes.
Wow, a self-perpetuating cycle of damage. That's a much more dynamic and frankly damaging picture than just an energy leak.
Absolutely. It provides a much deeper molecular understanding of how beta cells get progressively damaged in T2DM.
So understanding this cycle is one thing. How did they actually try to break it? This must be where the therapy comes in.
This is where the innovation really shines, building directly on that 2007 insight about UCP2 being a target. To inhibit UCP2 activity in vivo, in a living system, they designed a tailored nanomedicine. They called it Mito-G.
Mito-G. Okay, what is it?
It's pretty remarkable engineering. It's a negatively charged ultra-small nanoparticle, only about 5 nanometers across. They synthesized it by polymerizing Genipin, which was known way back as a potent UCP2 inhibitor, but had problems with glycine.
Right, Genipin. I remember you mentioning it couldn't really be used before. Why is Mito-G the breakthrough then? It solves the delivery problem.
Exactly. This tackles a nearly 20-year bottleneck. Genipin itself worked great in a dish, in vitro, but you couldn't use it in animals or people, in vivo. It had issues with cross-linking proteins randomly, causing side effects, and it just couldn't get to the pancreatic islets effectively. It was like having the key but not being able to read the lock.
So how does Mito-G reach the lock?
It uses what they call sequential targeting. It's a two-step process. First, its tiny size and negative charge allow it to slip through unique gaps, fenestrations, in the capillaries supplying the pancreatic islets. These gaps were about 6 to 8 nanometers, so Mito-G just fits.
Like a specific key for the islet door?
Sort of, yeah. And then, once it's inside the islet near the beta cells, it has a natural high affinity for the mitochondria themselves. It specifically binds to proteins on the mitochondrial outer membrane, like TOM20, which acts as an entry gate. So islet first, then mitochondria. Incredible specificity.
That really is precision medicine on a nanoscale.
Ah.
So, okay, it gets there. What were the actual therapeutic effects when they tested Mito-G in their diabetes models?
The results were genuinely impressive. In both the high-fat diet streptozotocin mice and the genetic DBDB mice models of T2DM, Mito-G effectively eliminated the beta cell autoinflammation, not just reduced, eliminated.
Wow.
This led to fasting blood glucose levels returning to normal within about a week at the higher doses. They also saw significant improvements in glucose tolerance and insulin tolerance tests, and critically, serum HbA1c levels, that long-term marker of blood sugar control, were significantly reduced.
What about the beta cells themselves? Were they recovering from all that stress and damage?
Yes, that was another striking finding. Mito-G treatment led to significant restoration of islets that were previously atrophic, shrunken, and deformed. They saw an increase in the total beta cell mass and a healthier ratio of beta cells to alpha cells, which is crucial for balanced hormone release. And functionally, it improved insulin synthesis. Serum insulin levels went up, and the expression levels of key genes needed for making insulin transcription factors like NURD1, MAFA, PDX1 were all boosted. It wasn't just stopping the damage, it was enabling repair and recovery.
So it broke the inflammatory cycle and helped rebuild. Did it have other protective effects beyond that main loop?
It did, yes. It
Yeah.
seemed to have multiple beneficial actions. It protected the mitochondrial membrane, potential essential for energy production, and helped restore ATP levels. It also inhibited the opening of something called the mitochondrial permeability transition pore, MPTP,
Yeah.
which is often a point of no return leading to cell death. And this reduced the harmful release of that mtDNA that fuels the sexting pathway.
Ah, so it hit the cycle at multiple points.
Seems like it. Plus, Mito-G itself showed some inherent antioxidant capacity, helping to scavenge damaging reactive oxygen species, or ROS. And finally, it significantly reduced beta cell death and also alleviated stress in another cellular compartment, the endoplasmic reticulum, ERS, which is also heavily involved in insulin production and often gets overloaded in diabetes. So a really multi-pronged approach built into one molecule.
That sounds incredibly comprehensive. How did its effectiveness stack up against, say, metformin, a standard treatment we use now?
Well, remarkably, in several key aspects, Mito-G actually demonstrated superior therapeutic effects compared to metformin in these animal models. Particularly notable was its ability to prevent beta cell exhaustion over the longer term and maintain higher serum insulin levels, suggesting it might preserve beta cell function better. And that highly specific targeting to the pancreatic islets with low distribution elsewhere suggests it could potentially have a much cleaner side effect profile than drugs that act more broadly throughout the body.
So let's sum this journey up. We started with the 2007 paper, giving us that first crucial map, identifying UCP2 as a player and a potential target. Then acknowledging that while the scientific conversation moves on, and sometimes those foundational links aren't always explicitly drawn in the very latest work.
Right, which is unfortunate, but happens.
The 2025 study we focused on today really brings that story full circle in a powerful way. It takes the initial UCP2 observation, dives deep into the dynamic inflammatory mechanisms involving mtDNA and SDing, and culminates in a novel, highly targeted nanomedicine, Mito-G, that addresses that mechanism directly.
Exactly, it's a massive leap. From understanding a single protein's basic role to designing a sophisticated nanomedicine that can navigate the body, find specific cells, enter specific organelles within those cells, and precisely intervene in a complex disease-driving cycle. It just brilliantly highlights the power of persistent scientific inquiry coupled with really innovative bioengineering to tackle these tough chronic diseases.
It absolutely does. This journey from identifying a protein target nearly 2 decades ago to designing a nanomedicine that precisely hits a complex inflammatory loop inside beta cells, it just opens up so many possibilities. Not just for diabetes, you have to think, but maybe for other diseases where mitochondria or inflammation are central players. It really makes you wonder, doesn't it? With this kind of precision becoming possible, what other chronic conditions might we be looking at completely differently, maybe even overcoming in the next 10 or 20 years? Well, that's definitely food for thought. Thanks for joining me today on the SANS Science Experience podcast. If you enjoyed this deep dive, please remember to like, subscribe, and share it. It truly helps us grow this community. This was the SANS Science Experience. See you next time.